Welcome to Biology of Marine Mammals. I'm Dr. Leslie Williams, and this lecture covers the very fascinating property known as echolocation. So echolocation is a feature almost exclusive to the Odontocetes. So we'll start by looking at what is echolocation, what is the purpose of echolocation, then we'll move on to the anatomical structures associated with the ability to echolocate, and finally we'll take a look at how to map an image and get a better resolution. Echolocation was first actually discovered in bats in 1938. It is the production of a sound with the intent of having it bounce back off an object. So the word echo refers to what you might think of as an echo, where you say a sound, it goes and bounces off, let's say an empty room or a big wall, and you hear it come back to you. Now, certain animals have the ability to hone this property, actually create sound with the sole intention of having it bounce back so that they can locate where an object is, the approximate shape of an object, and how far away that object might be. So as I mentioned earlier, this was first shown to be proven in bats in 1938, and in 1960 they were able to show that it also existed for many of the Odontocetes, including the Delphinidae, or the dolphins. So what is echolocation? Echolocation is really actually used to get a spatial map of objects. For dolphins, this is going to be prey items and probably their primary mechanism of finding prey, as well as just a major sensory adaptation for their environment. So for example, if you have a dolphin in a tank, they may not actually be able to see the clear glass in front of them, but they know it's there because of this echolocation. So it really is their primary sense to identify what's around them and locate their prey. We're going to come over here to this dot cam and take a look at how echolocation might actually help them distinguish between a small distant object and a larger close-up object. So right here I have this dolphin, and I've got a small distant object over there. They are going to emit sound. This sound is going to be in the series of clicks. So these sound waves are going to be funneled usually straight ahead, like so. And of course, sound travels at a fixed speed in water. And what's going to happen is all of these sound waves are going to keep going out, but this one is going to hit this small object here and bounce back. Now, this bouncing back here is going to actually be picked up by their hearing organs and sent to their brain. The way the brain is going to interpret this is that there was a single bounce back from this direction, so there's something over there, and it's fairly small. Otherwise, we would have had a bounce back from here and here and here, etc. But because it's just this one bounce back, there's a small object, and because it took so much time for that bounce back to occur, we know that it's fairly far away. All right, what happens when we have a large close-up object? Again, same principle, but look at what happens here. It doesn't take nearly as long for those sound waves to reach that large object, and it bounces back from multiple positions. So the dolphin is going to receive a signal here, here, and of course coming back over here as well, which tells us, hey, there's something right in front of me, and it's pretty large because I'm getting signals from multiple avenues here from these sound waves that tell me that it spans this entire distance, such as, for an example, an aquarium wall. Okay, so now that we've actually looked at how echolocation works, let's actually look at some of the anatomical features associated with echolocation. The first thing that we need to look at is sound production. So again, the dolphins are going to be emitting a particular sound, and it's clicks, a series of clicks, which can be over 1,000 clicks per second. So they can actually emit a lot of them in a very short amount of time. These clicks, as we now know, are produced not in the larynx, as most sound is produced for mammals, but in something different called the phonic lips. The phonic lips are going to be what almost seems to be like vocal cords, but they're located farther up in the nasal cavity. On this skull right here that's in front of me, you can actually see this is the nasal cavity, and the location of those phonic lips would be right about here, just below what would be the blowhole. Now, right in front here is kind of a fat-rich area called the melon, and that melon ends up being what's called an acoustic lens. A lens is going to be something that helps funnel some type of wave. In this case, it's going to funnel sound wave, hence the name acoustic. So it works very similar to the way our lenses work in our eyes to help focus light waves so that we can see better, but this is going to help focus the clicks that are being produced by these phonic lips out in front of them in kind of a directed beam. So what about the reception of this sound? Assuming that you do send out a click and it gets bounced back to you, how do you pick up on that click? Well, as we learned in a previous lecture, it turns out that in order to hear anything, you need to get your auditory ossicles or your ear bones rocking, which then transmit that information into the cochlea, which is sent to the brain. However, as we also learned, odontocetes do not use the normal ears that humans and even other pinnipeds and possibly other marine mammals use. They don't have a pinna, they don't have an external acoustic meatus. Instead, they collect the information in their jaw. And when they have that jaw is hooked up to a special a special bone called the petrotympanic bone, which is completely insulated in air, so it doesn't actually receive vibrations from anywhere else in the skull. It has to specifically come from the jaw. So again, we have this fatty channel here in the jaw, and what we're going to do is we're going to take that jaw and we're going to collect that energy and move it down the fatty channel here, all the way back to this petrotympanic bone, which contains the auditory ossicles. So the site of collection is really this fatty channel, which is on the inside of the lower jaw or the mandible. So they produce clicks, we send them out through the melon, and they bounce off an object or echo, and they're collected in this fatty channel, which then is transmitted to this petrotympanic bone containing the auditory ossicles. So once they're received, how does the brain make sense of that? Well, we can actually use a series of clicks to kind of create a spatial map. This is actually very similar to the way ultrasounds work. We use diagnostic ultrasounds to help identify things like the babies inside, or the fetus inside of a mother. We can also use an ultrasound to identify a heartbeat. And these are using a series of pulse clicks that bounce back and therefore create kind of a spatial image. So we're going to take a look at how that works. But I do want you to know that before we move on and look at how this works, that when you're trying to map images, you do need to be conscious of the pitch and the frequency of the clicks generated. So remember, a low-pitched click is going to be one where the sound waves or the peaks are very far apart, and a high-pitched wave is going to be one where the peaks are very, very close together. The low-pitched waves, remember, are going to travel a lot farther in the water, where the high-pitched waves don't travel very far, but they end up, as we'll see, having a lot better resolution. So let's take a look at how that works. When a dolphin or any odontoceti that's going to use some type of echolocation to sense its environment actually starts sensing, it's going to start with what's called exploratory clicks. The exploratory clicks are going to be these low-pitched and infrequent clicks. So maybe something like click, 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 and they're going to be low-pitched, and basically they're out to sense what's out in front of me long range. What am I looking at here? If you identify something that might be a potential prey item like a fish and you're going to hone in on it, they actually change the frequency and the pitch of their clicks. They will start using much higher pitched clicks, ones that move to the ultrasonic range. Remember, ultrasonic means beyond our hearing. So these are super high frequency clicks that are very, very tight waves, and then they're also very frequent. So instead of generating them, click, click, click. It's more like click, 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 click. And that's where you get the really fast set of emission to help you get a much higher resolution of what's in front of you so you can accurately locate that prey and grab it. Remember, odontoceti don't rely on their eyesight very much. In fact, many of the river dolphins are almost completely blind, and they rely exclusively on the spatial mapping to identify the exact location of a fish, especially as, of course, that fish is trying to get away. And so they need to be able to grab it, locate it, and grab it with great accuracy, or they're going to starve to death. So let's take a look at how these pitches are going to differ through exploration versus locating and identifying a prey item. All right, in this image here, what I have a dolphin here that's going to identify a fish that's far away. Now, as part of the exploratory clicks, it's going to send out a series of waves that are going to be low pitched. So again, these are going to be peaks and valleys are far away. And again, you're trading off the idea that you get long range at the expense of resolution. Now, as I've drawn the waves here, none of them actually identify that there's a fish involved. So as they move closer, they might start doing tighter waves like this. And then suddenly, two of them are going to pick up on the fish. Now, as those waves end up bouncing back, so this one and this one bounce back, here's what the dolphin is actually going to image. Something is over here, here, and here. We don't know exactly what's over there, but we know something is there and there, and it's not anywhere else in my spatial map. Now, as I get closer to this fish, I'm going to go ahead and start sending out much tighter waves at much higher frequency. These tighter waves, as you can see as I'm drawing here, they're very, very tight, are going to bounce back with much better resolution, allowing me to ascertain as a dolphin that straight ahead there's nothing, but there seems to be something here, 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 and here, and you can almost just connect the dots and say, okay, that's most likely going to be a fish. And so not only do they know that the approximate length of the object, because of the indentations and the reflections being a little different, it takes longer for this wave to bounce back than the wave that hits the tail. They know that it looks like this as well. They don't know what it looks like on the other side, but through experience and behavior, they can pretty much guess, okay, this is a fish, and if it's moving, it's almost definitely a fish, and we want to turn in that direction and try to capture that. So again, with the resolution, we're looking at using infrequent long-range clicks for exploration that have a long range before they dissipate, but the problem is that your resolution is poor. This is poor resolution, where when you get close to an object, you're going to use many more frequent high-pitched clicks, which give you a much greater resolution of what's right in front of you, needed to identify that prey item and capture it. And with that, I will see you next time.